Hello, thank you for joining us today as I present an introduction to Illumina's BaseSpace Correlation Engine and invite you to join our efforts in unlocking knowledge in SARS-CoV-2 host infection data. The agenda will begin with several slides to set the stage for what Correlation Engine is, how it fits into Illumina offerings, and where to request free access to the system. I will show how omics-scale data curation and aggregation from public resources can produce novel insights independent of or alongside integration with your own private data. After the slides, I will present Correlation Engine Live to highlight the basic frameworks and to dive into use cases derived from transcriptomic data generated by the tenOever lab and an Emory study looking for RNA blood marker associations to COVID-19 severity. I will touch upon pathway enrichment, drug intervention, correlations to single-cell RNA-seq studies, and a crowdsourcing collaboration that developed temporal RNA metasignatures during the course of SARS-CoV-2 infection in mouse lung. Illumina is known for sequencing and striving to improve human health by unlocking the power of the genome, and has contributed greatly to understanding the evolution of SARS-CoV-2 variants. An extension to the management, preparation, sequencing, and analysis of samples is facilitating collaboration through software solutions. Our software tools support the fight against SARS-CoV-2. Last year, Illumina released an instance of Correlation Engine freely available to scientists interested in coronavirus host response, with the mission to put observations into context, thereby accelerating research by staying current and pushing knowledge boundaries with integration and collaboration. As the COVID-19 pandemic has not subsided, we'd like to engage more researchers. Dr. Michael Edwards put it this way: BaseSpace Correlation Engine goes beyond simple text mining for pathways and allows me to compare my system to actual experimental results from thousands of curated studies in their repository. I can use it to find drugs or compounds that impact my genes of interest in the Pharmaco Atlas, look for similar phenotypes in the Disease Atlas, or even generate my own data sets on the fly using the meta-analysis function. It has become an indispensable tool in my bioinformatics arsenal. The link at the bottom is where you can request free access to this instance. Before going into details on the functionalities, I like to show an example end result. This screenshot is from the Pharmaco Atlas application. I queried an RNA expression signature from the tenOever lab on primary human bronchial epithelial cells, NHBE cells, infected with a particular variant of SARS-CoV-2 for 24 hours versus mock-infected cells. 597 genes were differentially expressed. Correlating this experimental signature to all signatures in the system that compared compound treatments to controls generated a rank-ordered list of 389 compounds with a negative correlation. That suggests certain disease-related dysregulation may be reversed. The top 14 drugs are shown here. It's quite spectacular. I overlaid annotations of compounds with their mechanistic targets to see the diversity of action. One thing we know about SARS-CoV-2 is that it's a large virus that disrupts a wide, complex breadth of host biology. The world needs your help in making sense of it. Several compounds discovered here are currently in clinical trials to treat COVID-19, like the tumor necrosis factor TNF inhibitors adalimumab and infliximab. We also see a couple of aurora kinase inhibitors known to interfere with mitosis. There are 2 IKKB inhibitors in the top 4 that would be interesting to understand better given the gene's role as a transcriptional regulator of immune response. From this web page, one can drill in further to better understand the underlying disease and pathway activities reversed by the drugs. I'll show you this page when I go live into Correlation Engine. To summarize, there are ready-to-use omics-scale experimental comparisons that one can cross-examine without needing deep computational skills. Curate, aggregate, correlate, and integrate is a sort of mantra we use to set a vision of what Correlation Engine is. Correlation Engine has a large collection of highly curated genomic data that has been pre-processed, normalized, and ontologically tagged. It's an easy-to-use web platform for exploring billions of associations and correlations with private and public data. It's built on big data frameworks, enabling secure collaboration and data sharing. The major applications are shown on the right. The Curated Studies App is the central repository of fully processed omics-scale studies. The Body Atlas addresses what tissues, cell types, and cell lines is my gene or gene set expressed in. The Disease, Pharmaco, Knockdown Atlases, and the Genetic Markers app ranks results for phenotypes, compounds, or genes correlated to a queried gene or gene set. There's a Pathway Enrichment app. There are powerful literature and clinical trials text-based apps that complement the experimental data-driven results one discovers in the other applications. And finally, my favorite, the Metaanalysis App that allows you to combine many signatures across experimental designs, molecular data types, and species to gain novel insight. We'll cover many of these in the demo. Correlation Engine empowers a broad range of scientists with access to public and one's private institutional data. Decision support for biomarker validation is backed by the large collection of highly curated genomic data. Correlations across molecular data types and phenotypes enable signature development of distinctive profiles. There are applications for compound, target, and pathway mechanism of action with combined cross-species analysis. For command line experts, there's a comprehensive API suite for integrations to and from internal resources. Though we remain a bit stealthy, there are over 650 primary research articles that cite Correlation Engine, spanning an inspiring breadth of biological, translational, and clinical science. I'm aware of 5 COVID-19 articles that use and cite Correlation Engine. Public content is added continuously to Correlation Engine by our curators. There are now over 22,300 curated studies containing over 231,000 molecular lists. I'll show the curation process in a couple of slides. The 2 screenshots here are from the Curated Studies app. On the top, you see the different molecular data types, like RNA expression, SNP-GWAS, DNA copy number, DNA methylation, histone modification, somatic mutation, and microRNA. The values in parentheses are the number of curated studies on July 1st. Around 50 new curated studies are added every month. The data types cover results from different technology platforms. For instance, there's over 19,000 RNA expression studies derived from various providers' arrays, and there's over 3,400 RNA-seq studies. The bottom screenshot shows the species covered. Most are from human, mouse, and rat. Ortholog gene clusters were computed that enable cross-species comparisons. For instance, gene knockout signatures in mouse can be compared to human disease signatures to help elucidate dysregulated molecular mechanisms. Correlation Engine is a hypothesis-free, data-driven environment. It seeks to capture as much biology as possible, so query results draw from broad and unbiased content. These pie charts show high-level coverage of the omics-scale curated studies and biosets by research area. Biosets are in essence experimental signatures derived from available samples in a study. You see, oncology comprises about 30% of studies in biosets. Stem cells differentiation and development, 16% of studies. Metabolic disorders, 12%. Immune response plus injury and inflammation, 20%. Neurology, 6%. Cardiovascular, 3%. This is a reasonable reflection of what exists in the public domain. Curated studies, on average, contain 6 biosets based on available pairwise experimental comparisons. Curated studies now cover over 6,200 phenotypes, 5,300 perturbed genes, and 4,800 compounds. There's a lot to work with. This schematic shows the Curators Workflow for turning data sets into processed biosets. Curators start with a publicly available repository of omics data in good annotation, like Gene Expression Omnibus, Array Express, and others. They review the sample types, do pairwise test versus control statistical analyses, and quality control, followed by some filtering on significance to create biosets. A simple curated study may have just one bioset, like disease versus normal, or treatment versus control, or knockout versus wild type. Others can have dozens or even thousands of biosets, like the Broad Connectivity Map, CMAP 2.0 study, with 3,442 biosets derived from 1,145 compounds tested in 3 cell lines. Next, our curators add metadata and ontologically tag each bioset with the essence of the comparison. We'll tag with the disease under investigation and the tissue, organ, or cell type. If a compound was used, then a compound tag will be added. If a gene perturbation was studied, then the gene and mode of perturbation tags will be added. Experimental design and other tags are also applied. These tags are used intelligently in apps like the Disease, Pharmaco, and Knockdown Atlases. This schematic depicts the correlation process. You can consider Correlation Engine in many senses an advanced search engine or adaptive learning machine. You can search by a gene, disease, compound, study ID, or as shown here, a ranked list of genes. For RNA expression, we use absolute fold change values. The gene with the highest absolute fold change is given a top rank of 1. For genome-wide association studies, the lowest p-value is given a rank of 1. Whether this is a list that you import or our curators derive from public sources, this ranked list is correlated to all ranked lists in your system, public and your own private ones, using the running Fisher enrichment algorithm described in our PLOS 2010 article. The first result is a ranked list of correlated biosets within studies. Based on the bioset correlation scoring depicted in the bottom graphic, each bioset is broken up into directional sets that contain the significantly up or down regulated genes along with gene ranks. Enrichment p-values are then determined in all directional combinations, which results in an overlap p-value for the comparison. This is automatically done between the queried bioset and the 140,000 other biosets in the system. From that, the lowest overlap p-value drives the best comparison to the top, with all rank ordered results below that one. It's not unusual to see thousands of ranked studies. A tag-based meta-analysis is subsequently run to make data-driven calls on what the most highly correlated diseases, compounds, and genetic perturbations are, and presented in the Disease Atlas, correlations to disease versus normal biosets, the Pharmaco Atlas, correlations to treated versus untreated biosets, and the Knockdown Atlas, correlations to genetic perturbation versus control biosets. Several other slides have been made available with this webinar. But for now, I want to dive into Correlation Engine Live to demonstrate the interactivity and stimulate thoughts on how you may use it. We're now live in the home page of the COVID-19 domain. For the general frameworks, you see the dashboard application icons on the top. Above that, there's short interactive help tutorials that walk you through examples across the applications. You'll see contextual help and specific FAQs throughout the system. The search box has examples listed below it, and the main action occurs in the central screen. There are facets on the left for importing and getting to your data, bookmarks, and collaborations. Let's look at curated studies. When going into a dashboard app without a query, quick start directions are presented. I'll open all curated studies. We see a chronological list of curated studies with species and experimental data type, bioset number, and links to full study details. There are over 22,827 total studies as of today. You see filters for the various organisms and data types that I showed in the slides. I'll query coronavirus infection. As I start typing, you'll see autocomplete vocabulary services that help to find the right concept. We now see 61 studies across 4 different species. As I scroll down and scan the study names, we see a wide range of viral types and variants, tissues, cell lines, treatments, gene knockouts, and more. After you've registered, you'll see the Collaborations app, accessible from the upper right facet. Clicking there, you'll see we created an open group called COVID-19 Community. In this group, I aggregated curated studies for coronavirus types, SARS-CoV-2, SARS-CoV-1, MERS, and other coronaviruses, into separate project folders to simplify location and sharing of studies. I also added several GWAS, DNA methylation, proteomics, and single-cell RNA-seq studies from tables available in publications. Let's look at the SARS-CoV-2 curated studies. The first curated study looked at RNA expression in lung cancer cells transfected with ACE2 and pancreatic organoids infected with SARS-CoV-2 and treated with 6 different drugs. The second study investigated RNA from COVID-19 lung biopsies and lung organoids infected with SARS-CoV-2 and treated with imatinib, a clinically approved tyrosine kinase inhibitor for certain cancers. The third study measured peripheral blood mononuclear cells of patients across a spectrum of severity presentations. We'll come back to this one from Emory University study in more depth. But you see Bioset signatures for the 4 COVID-19 severity level versus PBMCs from healthy subjects. As severity rises from convalescent to moderate to severe to intensive care, the number of significant differentially expressed genes increases from just 3 genes to 3,400 to 4,400 to 5,600. In essence, more biology is triggered as severity increases. Gaining a better handle on genes and pathways that change can yield insight in how to intervene in a stage-wise fashion to favorably change the course of disease progression. As we scroll down, we readily see a wide variety of studies and experimental designs. Our curators make the studies readable. As this one here, lungs from postmortem patients deceased due to SARS-CoV-2. Let's take a look at the full study details from the Icahn School of Medicine at Mount Sinai, a 10-hour study. The study details comes from the primary data source, gene expression omnibus in this case. If you know the source ID of a study you're interested in, then it can be queried to get right to it. If it's not in Correlation Engine, then you can request curation of it. There's a link to the research article, and we see samples were run on an Illumina NextSeq 500. The Biosets tab shows our curators were able to generate 15 pairwise test versus control biosets from available samples. We see biosets from NHBE cells, primary normal human bronchial epithelial cells, CALU3 cells, A549 cells with or without ACE2 transduced. We see biosets from infections by particular variants of SARS-CoV-2, influenza virus, respiratory syncytial virus, and human parainfluenza virus. With these, one could compare transcriptional effects between different respiratory viruses. For instance, what's the same and what's different? There's another comparison of lung biopsies from post-mortem of subject with COVID-19 versus healthy. It'd be interesting to look at the extent to which it may validate the independent experiment from patients deceased due to SARS-CoV-2 and the progression biomarkers from the severity biosets that we saw a moment ago. Let's look more closely at the first bioset. NHBE cells infected for 24 hours with the SARS-CoV-2 Washington variant at a multiplicity of infection of 2 versus mock-infected NHBE cells. One can open the View Bioset Details to see how our curators processed broad data. The list of genes is here. The top gene, CSF3, has the greatest absolute fold change, up 8.079 fold, and has a rank of 1. Subsequent genes are ranked by absolute fold change. The P values and Q values and test and control expression values are also listed. Below the list, you see the tags that our curators have applied. For biodesign, there's the tag disease versus normal. Biosource is primary cells cultured. The phenotype is coronavirus infection. And there are tissue tags for bronchi and epithelial cells. You can export biosets for use outside of Correlation Engine. You'll see export buttons throughout the system. Pathway Enrichment app will correlate this ranked list to over 91,000 functionally related gene sets curated from expert resources. We call them biogroups. Sources include the gene ontology, microRNA targets from TargetScan, protein families from Interpro, and several broad MCDB categories. I generally like gene ontology for their logical organization of concepts. I'll check that box and apply filter. The p-value ranked results show inflammatory response, response to virus, cytokine activity, and when we open the leukocyte migration, as an example, we see the breakdown of up and down regulated genes in the bioset. Biogroups as functionally related gene sets do not have numerical data in and of themselves or directionality. 32 leukocyte migration genes were observed upregulated and 4 genes downregulated. You can export the table of genes in the figure for this result, or at the higher level, export results across all significant biogroups. Let's look at correlations of the SARS-CoV-2 bioset to all drug treatment versus controls in the Pharmaco Atlas. This app ranks compound tags into 4 categories: mechanism of action, physiological effects, therapeutic use, and toxicity. The compound category locator shows the category-specific child concepts. I often like to view by ranks. We see that most of the top compounds have positive correlations to the SARS-CoV infection bioset queried, meaning the compound stimulates similar activities as CoV-2 infection. As I'm interested in compounds that may reverse the expression of host infection genes, I can filter by negative correlation with query. Here we see the results that I showed in the slide. Anyone can drill into these to better understand mechanisms to aggregate disease. With the NHBE CoV-2 bioset still in play, I want to go to the Curated Studies app and have you recall the correlate slide I showed. The ranked list of 597 differentially expressed genes was correlated to the 140,000 other biosets in the system. And we see over 20,000 curated studies are ranked from top to bottom by highest bioset correlation. This is purely data-driven. Let's open the top study to see why this came to the top. In this independent study from Osaka University, we see the top bioset was also from NHBE cells infected with SARS-CoV-2. Clicking on it, we see the breakdown of up and down regulated genes in each bioset, a Venn diagram of the overlap, and a bar chart with directionality determined p-values. There are highly significant positive correlations. Genes up in our query are also up in the discovery bioset, and likewise for downregulated genes. Relatively few genes are observed as negatively correlated, and those genes are also low in the ranked lists. The p-values are not significant. The table shows the CSF3 gene was ranked number 1 in the comparison. Let's contrast that to the second bioset in the study. Where NHBE organoids were infected with SARS-CoV-2 and treated with Camostat. First thing we see is that there is a strong negative correlation. Genes that are upregulated in the disease versus normal bioset appear downregulated by Camostat. CSF3 is observed upregulated 8-fold in the queried infected versus mock-infected bioset, while Camostat treated versus untreated experiment shows CSF3 gene downregulated 2-fold. Camostat is a clinically proven inhibitor of TMPRSS2 protease that the SARS-CoV-2 spike protein uses alongside the ACE2 receptor to invade cells. The Metaanalysis App allows us to look at relationships across many biosets. I want to know how robust the Camostat effect is. To do this, I went back to the top and I'll add a query bioset by clicking on this blue filter. And then I'll go down to the other 2 biosets and click on the meta-analysis icon there. Now at the top in the meta-analysis application, we see 3. Let's click on that. I first come to the Setup tab. Let's look at the Gene Results tab. We see a ranked list of genes with differential expression micrographs, with a bar for each of the 3 biosets. The top gene had the highest ranking across all 3 biosets. A quick scan of the level 1 results shows the 2 infected versus uninfected biosets are different than the 3rd Camostat, treated versus untreated bioset. Several chemokine and interferon genes appear at the top, upregulated in the infected versus uninfected biosets, downregulated in Camostat-treated SARS-CoV-2 infection. And as I scroll down, we see this is also the case for a couple of genes like MAP7D2 and keratin 15, where the genes are downregulated in SARS-CoV-2 infection, but are reversed to up expression after the infection is treated with Camostat. There's also a Biogroup Results tab. In the next workflow, I want to go to the Quick View page and start with a gene-initiated query using AURKB as a couple of Aurora kinase inhibitors came up in the top Pharmaco Atlas hits for NHBE cell SARS-CoV-2 bioset. The Quick View app lists the top 5 concepts in all relevant apps. We see the Disease Atlas lists severe acute respiratory syndrome, SARS, as 5th in the data-driven list of phenotypes. The General Info tab shows aurora kinase B participates in the regulation of alignment and segregation of chromosomes during mitosis and meiosis through association with microtubules. The human AURKB gene sequence is highly conserved as it has orthologs to the mouse, rat, monkey, cow, gorilla, dog, and fish gene. Scrolling down, we see more information, like which gene ontology concepts is AURKB a member. We could explore the body atlas, disease, pharmaco, knockdown atlases, but in the interest of time, let's check the literature out. We see 1,538 literature results. Cite aurora kinase B and its synonyms. An interactive tag cloud enables filtering by co-citation terms, like compound or phenotype. These can be used as filters to find the articles we want to link out to and read. The Clinical Trials app has similar interactivity derived from nightly updates from clinicaltrials.gov. In Curated Studies, we see 7,891 correlative studies. When exporting the results, we see over 28,800 biosets observed AURKB as significant. That's 17 times more experimentally significant AURKB observations than PubMed articles. The point here is that Correlation Engine mines real experimental data, not just what authors write about a subset of genes in their omics-scale studies. Searching both literature and experimental findings are useful. Correlation Engine simplifies discovery and contextualization of biology. Now let's use the advanced filters to go to the collaboration projects and select the ones I aggregated for SARS-CoV-2 studies, as well as single-cell sequencing studies, and apply the filter. The top study is the Emory PBMC COVID-19 severity biosets I showed earlier. AURKB is up over 8-fold in COVID-19 moderate, severe, and ICU, yet not significant in convalescent versus healthy patients. The second study from Lau et al. Measured single-cell RNA levels of bronchoalveolar cell immune cells from COVID-19 patients. It reveals proliferating T cells as the major contributor to PBMC upregulation of AURKB. Along the 13 other bronchoalveolar alveolar immune single cell types did not significantly express AURKB. The fourth study also measured single cells from COVID-19 subjects and also indicates proliferating T cells as a contributor to PMCBC upregulation of AURKB, along with CD8 positive T cells and megakaryocytes. I'd like to end here, as I could run on forever. I did not get to show off the work I'm doing with my wisdom of crowd collaborators, generating SARS-CoV-1 mouse lung infected temporal metasignatures. Suffice it to say, the correlations and extensions to the human SARS-CoV-2 infection studies at bulk and single cell RNA expression levels are astounding. I'd be happy to meet again to discuss that with you. Please contact us to get you registered to join in the search to better understand the mechanisms and potential interventions to this devastating pandemic. If you have ongoing research, then you can quickly assess and validate in silico which directions your research heads and publish with more confidence. Thank you.